presentations. I will be focusing on inflammatory bowel diseases that are like this. The main forms of the diseases, Crohn's disease and ulcerative colitis, that, as you may know, the Crohn's disease mainly affects any proportion of the intestine, principally localized in the ileum and colon. It's a discontinuous disease, and the process of the inflammation is transmural. The opposite to ulcerative colitis, that the colon and rectum is mainly restricted the inflammation, has a continuous extension, and the process of the inflammation is restricted to the mucosa and submucosa. Up to date, the prevalence in developed countries is 1 in 1,000. There are many clinical phenotypes. It's a quite heterogeneous diseases, are multifactorial and have no cure. Has been shown that 20 to 80% of the patients require surgery. It's a chronic disease and made debilitating, and has an increased risk of colon cancer development. So, as mentioned recently, these diseases are multifactorial, and this diagram shows that mainly 3 factors contribute to the etiology of these diseases, like genetic factors, the immune response, the environmental factors. But recently, and as was told in the 2 previous talks by Dr. Good and the previous talk too, that the barrier function is in these diseases is quite important. Also the microflora, and the now came to date the stress of the endothelial reticulum that was told by in the first day that the autophagy is also important. So one of these main factors that contribute to the diseases are the immune response and the genetic factors. So I will focus most of the talk in relation to the immune response and some factors that can be impaired in these diseases. So as a general point of view of how immune responses are triggered, we classically see that infection is the main trigger of innate and adaptive immune system as a defense mechanism, triggered by pathogen-associated molecular patterns present in microbial, but came to date that also tissue damage and cell stress produce some DAMs that are called damage-associated molecular patterns that also can activate signaling pathways of inflammation, maybe mainly through toll-like receptors, and recently shown by this receptor I will be focused in this talk, that is ST2, that is driven by the DAMP or the alarming IL-33 or interleukin-33. It seems that this sterile inflammation pathway, absence of infection, can drive the tissue repair and the adaptation to stress. But if any of these steps are altered, as occurs in inflammatory bowel disease, many inflammatory processes can be linked to them, such as autoimmune, sepsis, and fibrosis. So let me briefly tell you that the characteristic of the alarmins or danger-associated molecular patterns are endogenous molecules, are produced by our cells. They are rapidly released following a cell death, but not by apoptotic cell death. They can recruit and activate receptor expressing cells of the innate immune system and promote adaptive and immunity responses. So the role of this molecule has been shown to restore homeostasis and promote tissue reconstruction after direct insult or secondary inflammatory effects. So this table briefly shows some examples of these molecules, and the interleukin-33 together with HMGB1, that is a chromatin-associated protein, heat shock protein, defensins, is also uric acid, are considered part of this family of alarmins that are classically secreted by an active process, and all of them has a role in inflammation and immunity. So this interleukin-33, as you can see, is expressed and present in many organs is quite ubiquitous. It's not only in the GI tract, but also in lung and the nervous system and in the heart. And mostly epithelial, endothelial, and immune cells express this cytokine. So this cytokine classically was shown to be secreted upon cell damage, as I previously mentioned, as is shown in this diagram, that, for example, epithelial damage that as occurs in these diseases, ulcerative colitis mainly, induce the release of this protein that can be processed by cleavage of, through protease in the extracellular medium and activate this activity, inducing the several functions. For example, recruit many inflammatory cells from the periphery, like T helper cells, neutrophils, eosinophils, mast cells, and macrophages, not only from the circulate bloodstream, but also resident cells can be activating, and as you can see through this receptor that is present in the membrane, and induce many chemokines and cytokines production, activation of these cells in the intestinal mucosa. So the trigger might be related to the damage of this epithelial layer, but also has been shown that endothelial cell damage can also release this cytokine. So what happens when this cytokine and this pathway at the cellular level is activated? As I said, the damage of the epithelial cell release the interleukin-33. This is in physiological conditions, and cells expressing the receptor that can detect these cytokines promote the release of many mediators, not only cytokines, lipid mediators, promoting an inflammatory response, recruiting cells, and activating a process of mucosa healing, as is said. This effect is driven, as I said, by the receptor that is anchored in the membrane, the ST2 receptor that generates a heterodimer with the interleukin-1 adapter molecule protein, ACP, and binds in the extracellular domain the interleukin-33, activating a signaling pathway of several steps I won't focus today, driving the activation of many genes, mainly inflammatory genes and gene of the repair pathway. However, there's evidence that a soluble variant of this receptor, as you see, is identical to the extracellular domain and has the same binding property of the ligand and can counteract the activation of this pathway and supposedly inhibit the inflammation, but maybe also the healing process. So here in more detail, the variants of this receptor, here we saw the membrane anchor receptor and the soluble variants, and is mainly produced by alternative splicing. That is a process at the messenger RNA. So what happened with this system during pathological conditions? It has been shown that in many diseases, not only infection diseases, mainly activated by parasites, but also in asthma, has been well characterized the activation of the system in allergy, cardiovascular disease, and also in arthritis and in central nervous system diseases. Few examples, like in lupus or rheumatoid arthritis, the soluble variants of the receptor I mentioned recently, ST2S, I will talk about ST2S along the whole talk, is increasing the serum of these pathologies. And for example, In asthma, patients under an attack, the levels of this molecule is significantly increased in the serum. Also has been done several animal models to study the role of this pathway in inflammation, for example, airway inflammation or collagen-induced arthritis. And we were interested around 5 years ago in the role of this pathway in the role of inflammatory bowel diseases and in the inflammation of the mucosa. So this is the picture of how the inflammation of the mucosa releases the IL-33 and promotes the recruitment of inflammatory cells and activation of inflammatory circle in the mucosa. So we saw that inactive, the expression levels of the cytokine and in the receptor in the intestinal mucosa changes with the activity state of the patient. This corresponds to immunohistochemistry of IL-33 in the top level of ulcerative colitis, inactive ulcerative colitis, and inactive. And you can see that in active patients, the levels of IL-33 is increase, as shown by the red dots. And in the receptor ST2 is shown in green in the lower panel. That is mainly increase in the active state of the disease. So we characterized in 2010 this pathway, and after that, another papers came up. We published in Inflammatory Bowel Disease Journal that the levels of the soluble variant of the receptor in the serum was highly expressed, or the levels are increased in Crohn's disease and ulcerative colitis patients. And when we characterize these patients, this cohort of patients, according to the active or inactive state, we found out that the active patients, the levels were significantly increased. And then in World Journal of Gastroenterology, we published that the nice correlation between the intestinal levels of ST2 and the serum levels of this marker nicely correlate in patients with the endoscopic score of 3. So this is shown in more detail here, that in 84 patients, we have a very nice correlation between active and inactive ulcerative colitis, with the suggesting that it's a potential biomarker. I am focused in this cartoon of the soluble variant of the receptor that is upregulated in this disease. So after that, we did a follow-up in 31 patients that were under different therapies to detect the potential viability of this molecule as a biomarker of activity. So what we did, we follow up for 12 months a group of patients where the baseline was patients in activity. So we detect the serum levels of this protein. We correlate with the clinical score, endoscopic score, intestinal content of the protein, and also with fecal calprotectin. That is a well-established marker of activation. And we, according to the therapy, because of all these patients were under different therapy, we took different samples at 1 month, 3 months, 6 or 12 months of the serum ST2, also correlate with the clinical score and with the calprotectin, and after 6 months, another colonoscopy was taken to detect and to see there is an improvement of the endoscopic score, and if it's possible to correlate with this potential marker. So what we found that this is just to remember, but most of you are very updated with the pharmacological therapy in these patients, that according to the activity, if they are severe, moderate, or mild, the kind of treatment is different, and it's important to induce or maintain remission, right? So this is the characteristic of the patients. In total, we recruit 27 because 4 patients didn't follow the protocol because it was 1 year, but 22 were ulcerative colitis, 5 were Crohn's disease, and these are the location of the disease in ulcerative colitis and Crohn's. The therapy was mainly a conventional therapy, and a few of them received biological therapy, anti-TNF-alpha. And we characterized the response of the treatment in group of responders and non-responders. Responders are those with their clinical score response to therapy. They have a reduction in the endoscopic and clinical scores, and declined the evaluation 9 of them. 8 patients were reactivated, and 14 finished the protocol with no reactivation. In the case of ulcerative colitis, and in the Crohn's disease were like even amount of patients. So what we see that the serum levels of ST2 around the 12 months, there's a nice decrease with the follow-up of the patients. You can see that the higher levels of the molecule in the plasma, but decrease along the follow-up. And we found a significantly different up to 1 month when they start responding to the treatment. But we didn't see the same pattern with the fecal calprotectin. You see, just only at the end of the follow-up, we could detect a difference. For the fecal calprotectin, we use an ELISA kit that gives quantitative levels of this protein. So we made an association analysis between the levels of ST2 and fetal calprotectin at the baseline when we start the follow-up, and 6 months in the group of responders and non-responders patients. And you can see that those that respond to the treatment, the levels of ST2 decrease, and not the same in the non-responders, but in the fecal calprotectin, we found a significant difference, but was not that strong as with the serum ST2. And also for the non-responder, we found a difference, a statistically difference that is not strong. However, we found that is not correlate what we see with the serum ST2 levels. So we found out that if we reanalyze this data according to those that respond in responders patients, these are non-responders because at this group, as you can see, that the levels did not decrease, so they restart in the protocol, and changing the therapy to biological therapy, the levels of ST2 nicely reduce as the same as fecal calprotectin. So when we correlate the levels of ST2 to the endoscopic score, we found that higher levels of ST2 are correlated to high endoscopic level as well as with the higher clinical scores. And the correlation with calprotectin is not that strong as we thought that would be. This data are under evaluation. So we did also immunohistochemistry to see if we can see the levels of ST2 in the tissue correlates with those or have the same trend as we saw in the periphery. And we saw that in the responders, the levels of ST2 were decreased, but not in the non-responders. And when they reactivate, the levels of the marker that is in the red dots decrease nicely. So ST2 can be we can now consider a biomarker of intestinal inflammation because high levels are indicative of an active disease. This molecule can predict the relapse and response to therapy. And ST2, like fetal calprotectin, can be considered as a useful biomarker in predicting the outcome of patients with IBD in the clinical practice. So after these observations, we have another cohort of patients that published in 2011 that we classified according to the different treatments, the levels of ST2. It was a bigger cohort of patients. And we found out that those receiving systemic corticoids, as you focus in this bar, the levels of this protein in the periphery are significantly higher of the soluble variant, again with the soluble variant. So it looks like the treatment has an effect on ST2, and mainly in those patients receiving corticosteroids. So we then subclassified those patients receiving corticosteroids in active and inactive. We previously shown that this marker can discriminate activity, but also if you focus on this part of the slide, the treatment with glucocorticoids in those patients in active state of the disease, that means score 2 or 3 of endoscopic score, the levels of ST2 are more high receptor, and in the presence of ulcerative morphisms require protein higher to those of the protein were significantly higher. Patients receive steroid treatment. So treatment of another, this one is the one that sequester and to many village. It has been shown that because this pathway has a role in Crohn's disease, and also because ulcerative colitis is more superficial. So,
I think that makes tremendous sense, and I think that could bear a lot of value from a clinical correlation and clinical treatment. A couple of other comments I have. One is that the difficulty you had in correlating the ST2 levels in the serum with the fecal calprotectin may be a problem with the fecal calprotectin assay. There's a lot of data that if you use a cutoff of 100 for fecal calprotectin, going from 100 to 300, the levels actually are not very reliable. But if you stratify the data to look at only those patients with a fecal calprotectin over 300, you may find a very much better correlation. So I would suggest that you look at your data, and you may be able to find a more linear relationship then, because that 100 to 300 range is a little unreliable.
Yeah, we start using these semi-quantitative calprotectin assays, and we found out what you said, that there's no correlation between when they have 1+ or 2+. So we decided to purchase a quantitative ELISA. So we have specific numbers in the levels of calprotectin. So we associate the levels we found with this assay to the ELISA of ST2. So so maybe we have to encourage the clinician to update the criteria, because they're quite fixed to this range in calprotectin, but maybe it's not that accurate or not that contributing to the reflection of what is happening in the mucosa.
Yeah, I think that there's been some publications to look at that. And my last question, and it's a comment really, and that is that the ulcerative colitis data that demonstrate that perhaps the steroid patients are expressing a higher ST2 level because of the genetic polymorphism may be part of a clinical problem that we are actually making some of these patients worse by persisting in using corticosteroids. There's a new model where we should be very aggressive about top-down therapy. And it may be that if we see someone after a relatively short period of time who does not respond to corticosteroids, we should immediately go to monoclonal antibodies or biologics, because it may be that the steroids are fomenting the disease and actually increasing the inflammatory process.
Yeah, I agree. And there are many, this is a newer mechanisms of how corticoids can affect the inflammatory pathway, but really we don't know what glucocorticoids are doing, which genes are regulating, and we try. I know that these patients doesn't receive the glucocorticoids for more than 14 weeks. So if they don't respond, they change to another treatment, of course. Yeah. Thank you so much.
Thank you.